Drodzy Państwo, nasz kolejny wykład wygłosi profesor Cristiny Sánchez. Będzie on się tyczył kanabinoidów i nowotworów. Naukowe odpowiedzi na najczęściej zadawane pytania. Powitajmy wielkimi brawami. Zapraszamy Panią na scenę.
Ja tylko jeszcze dodam, że już dzisiaj raz użyłem wyrażenia najwyższa półka światowa. Zdecydowanie to wyrażenie jest na miejscu w tym momencie także.
Hi, can you hear me? Yeah, I don't. Well, thank you very much for having me here. It's a pleasure to come to Poland for the first time. Beautiful country. And a special thanks to Bogdan. Thank you very much for giving me the opportunity to be here today. Well, I'm going to try to answer from a scientific point of view most of the questions that we receive in the lab. We are not doctors, we are not medical doctors, we are doctors in biochemistry, but we don't treat patients. Our patients are mice. So all the work I'm gonna talk about, except when I specifically mention that I'm talking about humans, is being carried out in preclinical models of cancer, in cancer cells cultured in plastic plates or in animal models of cancer. But what I'm gonna try to do today is to answer the most common questions that we receive from patients asking about cannabis for their condition, which is cancer. So, first of all, I'm going to make a brief introduction. I'm very sure that you have already gone through this with other speakers, but to me it's very important that you have very small pieces of information very clear to understand what I'm going to talk about today. And the first thing I want to remind you is that we are talking about a plant that produces hundreds of chemical compounds. Cannabis sativa, marijuana, produces hundreds of chemical compounds that we classified or that we group in different groups depending on their chemical structure. So we have compounds that we call alkaloids, flavonoids, terpenes that are present in other plants, but this specific plant produces a family of unique compounds only produced or mainly produced by this specific plant that are called cannabinoids. In this drawing, I have included 4 of them. We have identified almost 150 already, and the numbers are still growing. And they look alike. They have a similar shape, a similar form, and that's why they belong to the same chemical family, the chemical family we call cannabinoids. The most famous ones are this guy here, delta-9 tetrahydrocannabinol. I think it needs no introduction, and CBD. But please keep in mind that within the plant, there are many other compounds, cannabinoid compounds and non-cannabinoid compounds with potential therapeutic applications. And I want you to keep this idea in mind because I will present you some experiments later on showing that if we have to use a cannabis compound, it may be a better idea to use something that comes from the whole plant instead of a single molecule. So, as you all know, we have cannabinoid receptors in our cells. These are the molecular targets that are activated by cannabinoids when we incorporate cannabinoids into our body. And as for now, there are 2 cannabinoid receptors described. The first one was characterized in 1990, and it was called CB1 because it was the first one discovered. And it is present all over our body. It was firstly named central cannabinoid receptor because it's very, very abundant in the central nervous system, but today we know that it's expressed all over the body. And this is an image of the CB1 receptor expressed in the brain of a rat. And I'm choosing this image because it comes from the original article where the cannabinoid receptor expression pattern was described. And this is the only reason why I put this image. But if we do something similar in humans, we will have a very similar pattern of expression. And with this technique, what the researchers did was to stain in black the CB1 receptors. So where you see very intense black signal, what you are seeing is very high levels of CB1 receptors. And when you don't see a very intense black signal, what you are seeing is lack of expression of receptors. In other words, no CB1 receptors are present there. And this information is very, very useful to understand why cannabis does so many things in humans. For example, if we go through this image, the area of the brain that is more intensely stained is this part here. This is the cerebellum. This is part of the brain that it's right here in this part of the head. And this is in charge of controlling many, many functions, and one of them is the posture. So right now, you guys are sitting in your chairs, I'm standing here, and the fact that we are having these postures has to do with our endocannabinoid system working in that part of the brain and controlling our posture. And that's why when we use cannabis, that is affected. I would be having coordination issues here standing, and you probably will be having coordination issues as well if we use cannabis, because we are touching the cannabinoid receptors that are expressed in the cerebellum. If we move in this slide, for example, to this area, this is called the hippocampus. And the hippocampus is the part of the brain that is in charge, among many other things, of processes as learning, memory, stress. All these things are controlled in the brain by cannabinoids, and that's why when we use cannabinoids, when we smoke a joint or when we use a cannabis product, all these things are affected. If I use cannabis now, my ability to learn new things is going to be impacted, or my memory, my short-term memory is going to be impacted, and my feelings, my stress sensation, the stress I have now because I'm facing an audience, will be reduced. And this is possible because we have CB1 receptors in the part of the brain that controls these emotions or these physiological functions. So it's very important to know where do we have cannabinoid receptors and to understand which physiological functions are controlled by those areas, because that way we can see if we can use cannabinoids to treat pathologies or symptoms that are controlled by the brain in that specific area. And it's also very important to understand where we do not have cannabinoid receptors in the brain. And if you go to the image again, there is a part of the brain that is very, very light in color, meaning that has very, very little amount of CB1 receptors, which is this part here. See? Nothing to do with the black expression here. This lack of color means that over there, and that's the brain stem, that's where the this part, when it goes through the column, we don't have receptors there. We don't have CB1 receptors. And that part of the brain is the one that controls heart rate and breathing. And that's why we can die by overdosing with cannabinoids, because there are no targets in this part of the brain. So it is very important to understand where we do have CB1 receptors and also where we don't have CB1 receptors to understand how we can use cannabinoids to cure some diseases. Sorry for using the word cure, it was a mistake, for helping in some diseases, and what things can't happen by overdosing or by taking too much cannabis. So this is regarding CB1 receptor, which is the main responsible, for example, for the psychoactive effects of cannabinoids. And we do have another cannabinoid receptor that was discovered 3 years later, and it was named CB2 receptor. And this receptor in adult and healthy individuals is restricted to the immune system. We find it in the immune system and only in the immune system, with very few exceptions. And I want to stress out the words adult and healthy, because we are getting information showing that in certain pathologies, and cancer is one of them, the CB2 receptors appear in cells where they were not present when the individual was healthy. And also, the word adult refers to the fact that when we are not born yet, when we are fetuses, in our development, we do have CB2 receptors all over the place, and they disappear when we get to adulthood. So that's why I insisted in using these two words. So the take-home message is CB1 is all over the place, CB2 is restricted to the immune system unless you are developing or unless you have an issue, a health issue. And why do we have cannabinoid receptors? I always say the same thing, but I think it's very easy to understand. We don't have cannabinoid receptors in our cells because maybe one day we may want to smoke a joint. Nature doesn't work that way. Nature doesn't waste any energy in producing something that may or may not be used. If we have receptors, it's because we have something in our bodies that act on these receptors. In other words, we have cannabinoids inside our bodies. We produce cannabinoids ourselves. And these compounds are called endocannabinoids, meaning produced from within our bodies. And there are a number of them, but the most known ones are anandamide and 2-AG, 2-alkyl glycerol. In addition, we have molecules that produce cannabinoids when we need them. Cannabinoids and endocannabinoids are not there all the time. You have to think about endocannabinoids as adrenaline, for example. We need adrenaline, but we need it present in our bodies all the time. That will give us a heart attack. And with cannabinoids, it's the same. I'm not saying that cannabinoids will give us a heart attack. I mean that when we need it, we produce them. They are not there all the time. So we have proteins, a set of proteins that are in charge of producing cannabinoids when we need them, and also of degrading them, breaking them up when we don't need them anymore. So together, these cannabinoid receptors that I just mentioned, CB1 and CB2, the endocannabinoids, anandamide and 2-AG, the proteins that are in charge of synthesizing, producing cannabinoids, and the proteins that are in charge of degrading, breaking down the cannabinoids when we don't need them, all these things together is what we call the endocannabinoid system. And where do we have an endocannabinoid system? We have an endocannabinoid system basically in our cells. And what is the endocannabinoid system doing in these cells? Well, it is controlling many different cellular and physiological functions. You name it. This is just for examples, but if you think about a physiological process, any physiological process, the chances that the endocannabinoid system is involved in controlling it are very high. I just mentioned here for example, pain perception. We do have an endocannabinoid system in the areas of the brain and the spinal cord that are in charge of perceiving pain, of feeling pain. And that's why we can use cannabinoids to control pain responses. As you all know, cannabinoids are analgesic compounds, and they work as analgesic compounds because we do have an endogenous system in the areas of the brain that control pain perception. Cannabinoids control, for example, motor coordination because we have an endocannabinoid system in the areas of the brain that control motor coordination. And that's why we can use cannabinoid, for example, for multiple sclerosis to prevent or to ameliorate the motor component of the disease. The spasticity associated to the disease can be treated with cannabinoids because we have an endogenous system present in that part of the brain. The immune response, same thing. We can modulate the immune response with cannabinoids because we do have an endogenous system, an endocannabinoid system in immune cells, and so on. This is just a few examples. And what about cancer, which is the field I work in? Well, we can use cannabinoids as well because cancer cells have an endocannabinoid system. It's an endocannabinoid system that is altered, but it is present in the cells. So we can target the endocannabinoid system to get therapeutic responses. So in this slide, what I'm going to try to do is to introduce you to cancer. I'm sure everybody in the audience knows someone who's dealing with cancer, or there will be probably many cancer patients here. And part of it doesn't need any explanation, but there are things that we usually don't think about when we think about cancer patients, and I want you all to have them very present when I talk about cannabis. So in the top part of the slide, what I represent is how a cancer progresses. It's the disease per se, how it progresses. So when we think about cancer, we all imagine masses of cells growing without control. That's part of the process. And this is a consequence of mutations that are produced in some of our cells. So one of our cells mutate, and then it generates daughter cells that carry this mutation, and the daughter cells generate more daughter cells that have the same mutation, and we end up having tumoral masses. This is what we know about cancer. But cancer is also accompanied by two other processes that are less known but equally important. The first one is what we call angiogenesis, is the generation of new blood vessels. Right now, we all, and please don't get scared, we all have tumors right now, but they are very, very small, very, very small. And we have a very active immune system that get rid of these tumoral masses, so they are not a danger for us. And also, we don't have this angiogenesis process in progress. The angiogenesis is the generation of these new blood vessels that feed the tumor cells. All the cancer cells and all our cells receive the food from the blood vessels. So when we have a tumor this size and it needs to grow, it needs an extra blood supply. And this is the angiogenesis process. And at the latest stages of the disease, we have another process that is called metastasis, which is in most cases, unfortunately, the cause of death, not the primary tumor, where we have some of these cells, some of these tumoral cells that are growing, escaping from the tumoral mass, getting into the blood vessels, traveling through the circulation, and getting to new locations where they start growing again and they generate new tumoral masses. For example, breast cancer. The main tumoral mass, the primary mass, is produced in the brain, in the breast, but these cells travel and they produce metastases, for example, in the liver, in the brain, in the bones, and unfortunately, this is the cause of death, not the primary tumors. Dealing with primary tumors is in general easy, dealing with metastases is not. And that's why it is so important to have all these three steps always in mind. But this is not all that cancer patients have to deal with. They also have to deal, in most cases, with the side effects of the anti-tumor therapies they take, that you all know are extremely aggressive. I have included in this slide just a very few list with some of the most common side effects that these chemotherapies produce. Nausea, vomiting, lack of appetite, pain. For healthy people, these may be minor things, but for a cancer patient, suffering these things day by day, all the time, is really debilitating, and it diminishes their quality of life tremendously. And in some cases, all these things make them leave the treatments, the anti-cancer treatments, which really endanger their condition. And we don't have to forget either about the psychological aspects of the disease. These patients and their families suffer from anxiety, suffer from trouble sleeping, which again is redundant with a poor quality of life and a deterioration of their condition. So what I'm going to do now is try to, as I said before, answer the questions that we get from the patients. And the first one is, I have cancer, can cannabis help me? And the answer is yes, it's a definite yes. But let's go piece by piece in this slide. We do know from clinical and preclinical studies that cannabis can be used to treat the side effects of the anti-tumor treatments, for example, and the psychological aspects that come with the disease. They can prevent nausea and vomiting induced by chemotherapy. They can increase appetite. They can reduce pain perception, reduce anxiety, and can help sleeping. Last 20 years has nothing to do with this palliative side of the effects, but with the anti-tumoral effect per se. So I'm going to show you now pieces of evidence. It's less than 100% of what we know that I'm showing them to you to convince you that we have preclinical evidence, enough preclinical evidence supporting the use of these medicines as anti-tumoral agents. Let's focus first on the first part of the progression disease, the uncontrolled proliferation, uncontrolled cancer cell proliferation. So when you have a potential anti-tumor tool in your hands, the first thing you have to do is to test it in cell cultures. And that's what we did. And this picture was taken in the late 1990s. And these are glioblastoma cells Glioblastoma is probably the most aggressive brain tumor. And in the left-hand side, you can see how these cells grow in these petri dishes. And these black spots here are cell nuclei. So each of these spots is one cell. And this is how they look when we treat them with THC, with a small amount of THC. And as you can see, the picture is completely different. We have very few leaves remaining there, and they have a completely different shape and form. They are round, they are refringent, and they are detached from the plastic surface. All of them are signs of cell death. So THC is killing the cancer cells. We've seen this, these are glioblastoma cells, as I said before, but we've seen this in every type of model cancer type that we have studied. Not only us, but many other groups working on cancer. So when we got these positive results, we moved to a more physiological setting. And when we talk about more physiological settings in a biology lab, unfortunately, we're talking about mice. And this is the easiest model we can work with in cancer. What we do here is to inject cancer cells, the same human cancer cells I showed you before. We inject them underneath the skin of immune deficient mice. They have to be immune deficient in order not to reject the implant. So we injected these cells into the mice, and they generate these tumoral masses. This is a brain tumor here in the side of the animal. And then we divided the animals into groups. Half of them are treated with cannabinoids. In this case, it was THC, and the other half is not treated with cannabinoids. And as you can see, the animals that received THC treatment have smaller tumors, dramatically smaller tumors, than the animals that did not receive cannabinoid treatment. So when we surgically remove these tumoral masses, this would be this one, and this white one would be this other one. You can also see, you can still see the difference in size. It's very obvious, but you can hopefully see, another difference that has nothing to do with the size, but with the color. The one in the left is red, meaning that it's full of blood vessels, meaning that it's giving a lot of food to the tumoral cells. And the other one, on the contrary, is completely white. So no tumoral vessels are irrigating this growing mass. In other words, cannabinoids, THC, is impacting not only cell growth, but also angiogenesis. Of the mice, but in the of rats, and these are immunocompetent rats, so their immune system is working perfectly fine. So we injected the tumor cells in the brains of these animals, and this is an MRI scan, same time of analysis they do to patients, and the tumoral mass is this white signal here and here. And these pictures I just pointed were before THC treatment. And the pictures you have on the right-hand side, here and here, this is the very same rat after THC treatment. As you can see, the tumoral mass has disappeared, completely disappeared. This doesn't happen all the time, but in 30% of the animals, we saw complete eradication of the tumors. And in the others, we saw a slower growth of this tumoral mass. But again, this is very good news, very good results, but we don't get tumors because someone comes and injects us with some cancer cells. We generate these tumors spontaneously because we accumulate mutations in our cells. So we had access to a different model, in this case of breast cancer, where the tumors are generated spontaneously. We have to do a genetic manipulation when they are in the two-cell state phase of development. But the consequence is that when they reach adulthood, they start developing breast tumors, and they do it spontaneously. So what we observed in this case is that if we didn't treat the tumors, they grew exponentially. In these graphs, we represent tumor size, tumor volume, versus time. And as you can see, the tumors grow exponentially. And when we treated these animals, same animals, with cannabinoids, in this case THC, the tumor growth was significantly reduced. So again, we are seeing antitumoral responses, in this case in a model that is very, very similar to the human pathology. What about the second phase of the disease progression, angiogenesis? I have already shown you an image showing you that this process is blocked by cannabinoids. Remember the tumor that was black, red in one case and white in the other? This is another example of the same thing. In this case, we generated glioblastoma tumors, again brain tumors, in a rat, in a rat model of cancer, and also astrocytoma cells, another brain tumor, coming from human patients. And what we did in these experiments was, after the tumors were removed, we inject them with a blue dye. And this blue dye has a special characteristic. It only travels through the blood vessels. So if we inject this dye into a tumor and the tumor gets blue, it means that it's full of blood vessels that are carrying the dye that makes them blue. And if we inject the colorant, the dye, and the tumor doesn't get blue, it's because there is no blood vessels there. It's very easy and the results are very strong. So we treated this, in this case, we treated the animals either with a compound, a cannabinoid compound that activates CB2 receptors specifically. Not important, it's a cannabinoid anyway. And we saw, if you compare the top row with the bottom row, we observe, again, antitumoral responses. The tumors down here are always smaller than the tumors in the top, in both cases. And also, when we injected the dye, the blue dye, we observed that the tumors that did not receive cannabinoid treatment, the ones in here, this one and this one, they turned blue when we injected the dye, meaning that they had blood vessels inside. On the contrary, when we did the same thing with the tumors that had received cannabinoid treatment, they didn't turn blue. In other words, they didn't have blood vessels inside, which is again telling us that we are blocking not only tumor cell proliferation, but also angiogenesis. And interestingly, cannabinoids also block the last part, the most dangerous part of the disease progression, metastasis. And in this case, we used the breast cancer model I mentioned before. These animals not only generate breast tumors spontaneously, but they also generate metastases. Approximately 70%, a little less than 70% of the animals develop metastases in the lungs if we don't treat them with cannabinoids. And when we treat them with THC, this number goes to below 20%. So we are tackling the 3 stages of cancer progression, not just one. So what about humans? Because all I have shown you up to now comes from either cell cultures or from animal experiments. So what do we know about the anti-tumor effect of cannabinoids in humans? Well, the only clinical trial, high-quality clinical trial that has been conducted so far is this one that I summarize here. It was sponsored by GW Pharmaceuticals. It was performed and finished a couple of years ago in hospitals of the United Kingdom and Germany. And there were 2 experimental groups in this study. In one of them, patients received temozolomide. Temozolomide is the drug, the treatment glioblastoma patients receive, is their chemotherapy. And in this group, doctors combined temozolomide and the cannabis drug, Sativex, that has a mixture of THC and CBD. And in the other group, patients received temozolomide plus placebo. And the results have not been published yet. We are waiting for the publication of them. But the company had a press release in their website, and they say what I have summarized here. they say that the 1-year survival was increased in the patient population that received the double treatment. It was 53% for temozolomide-only patients, and it became 83% when patients also received the cannabis medicine. The median survival of these patients was roughly 1 year for those that didn't receive cannabinoid treatment, and it was increased to 550 days for those that received Sativex. And something that we consider very, very important is the number of patients that discontinued the treatment due to adverse side effects. And you see the number is identical in the placebo group and in the Sativex group. And this is suggesting that whatever made these patients leave the trial had nothing to do with the cannabis. It was 2 in a group of 9 and 2 in a group of 12. If we put that in percentage, it's even less in the population that received cannabis. So cannabis is not doing anything that is making the patients leave the study. And there were some adverse effects related to the use of cannabis. And they detected in 3 patients or more vomiting, dizziness, nausea, headache, and constipation. If there are cancer patients here or doctors that deal with cancer patients here, you all know that this is something we can tolerate way better than all the toxic effects that regular anti-cancer treatments produce. So then another question that we are often asked is if cannabis would work for any type of tumor. We've been working mostly in brain tumors and breast cancer during these last 2 decades, but other groups have been also working in this field. And the answer is, well, we don't know if they will work, but chances are that they will, because for all these types of tumors, we have seen anti-proliferative effects, cell-killing effects. The list is huge, the list is still increasing, and there is something, we suspect that there is something that all cancer cells share that we haven't identified yet, unfortunately, that makes them susceptible to be treated by cannabinoids, to be killed by cannabinoids. This is regarding anti-proliferative effects. For anti-angiogenic effects and anti-metastatic effects, the list is shorter, but not because some of these tumors do not show these responses, but only because these responses have not been studied in those other tumors. If I have to bet my money now, I would win for sure if I include these other type of tumors in here as well. So the lack of, or how short this list is has nothing to do with the responsiveness of the other types of tumors. So another question that is very important for patients and for doctors is cannabis safe? And I'm going to start answering this question from the lab, from the preclinical setting. And the answer is yes. In our hands, these compounds are very safe. So the same way we start cultures when we want to see efficacy of an anti-tumor compound, when we want to see safety, we start as well with cell cultures. We get when we do experiments in these cell cultures. In this case, we used a number of different cell lines. This will represent somehow different breast cancer patients, let's put it this way. And the blue and the green bars represent their cell viability. We want this bar to go down. Less cell viability means we are killing a lot of cells, so we want the bar to be as low as possible. And what you see here is that when we treat all the blue and green cancer cells, breast cancer cells with cannabinoids, the bars go down. All of them except the yellow one. And the difference between the yellow and the green and blue is that the yellow bar represents the response of non-tumoral breast cells. And we have observed this over and over and over when we study different types of tumors. If we work with breast cancer, we do experiments with normal, non-tumoral breast cells, and they don't die in response to cannabinoids. The tumoral equivalents die. The healthy equivalents don't die. If we do that with brain tumors, for example, if we are working with neuroblastoma. Cannabinoids kill neuroblastoma cells, but they don't kill neurons. If we work with hepatic tumors, for example, cannabinoids will kill the liver cancer cells, and they will not affect the hepatocytes. And this is a response that we have observed, I said before, in all types of tumors we have studied. Again, what happens in vivo, in animal experiments? What we do every time an in vivo experiment is, after taking the tumors from the animals, we send the rest of the organs to a pathologist to see if there is any kind of sign of damage anywhere. We send brain, lung, heart, liver, spleen, everything. And we have never ever seen any sign of toxicity in any of our animals. But in this case, we have some information coming from clinical studies performed in humans, suggesting that cannabis is a safe medicine for cancer patients. This is a summary of all the studies I found. This was updated in February, with cannabis medicines that are currently authorized in different countries. And 336 clinical trials have been completed already with Sativex, and none of them suggest that this medicine is any dangerous for a cancer patient. Marinol, which is THC, exactly the same thing. 107 studies, and none of them suggest that this could be dangerous for a cancer patient. And exactly the same with other compounds, including CBD. So we have hundreds and hundreds of clinical trials. This means thousands and thousands of patients in which cannabis medicines have produced no toxic effects. So another question that we are often asked is, what type of cannabis do I use? Once I have made the decision of incorporating cannabis into my therapies, what kind of cannabis do I use? And this question is very wide, but I'm going to focus on a specific sub-question. Is it better to use pure compounds, pure THC, pure CBD, or something that comes with the other components of the plant that I mentioned in the first slide? And that's why I wanted you to keep that in your minds. Well, to answer this question, we established a collaboration with this company, Zelda Therapeutics. One of the founders is sitting here and is going to take the stage right after me, Mara Gordon. And we compared the anti-tumor effect of pure THC that was provided by this company here with that of a THC-rich extract that is produced by this non-profit mutual benefit corporation in California, on Celdas. This is the test results of this extract. It has a known amount. The number is irrelevant. I'm showing you this just for you to keep in mind that we know how much THC this extract has. And we not only know the amount of THC, but also of the other components, terpene components, other cannabinoids that are present in this extract. And what we did was to test these two therapeutic options in models of the 3 different types of breast cancers that we have. We have a first type of breast cancer that is called ER-positive. ER stands for estrogen receptor and PR for progesterone receptor. We also call them hormone-sensitive tumors. And the treatment that these patients receive is, the goal of these treatments is basically shut down the estrogenic signaling. So stop the estrogens to activate the estrogen receptors. And we can do that by many different approaches. One of them would be removing the endogenous source of estrogen, the ovaries, or using drugs like tamoxifen or anastrozole that target somehow in different ways the estrogen receptors. We have a second type of breast cancer that is called HER2 positive. HER2 is a protein that is abnormally upregulated in these tumors. So we have way more amount of this protein than we need, and this causes this type of cancer. And we can target these tumors with medicines that are specifically designed to target this protein, HER2. Among them, there is trastuzumab, pertuzumab, and this molecule here, lapatinib, that I will mention in a few slides. And then we have a third type of breast cancer that is called triple negative, and this name comes from the lack of expression of either estrogen receptor, progesterone receptors, or HER2. That's why it's called triple negative. And since it doesn't have anything to target, these patients can have any specific medicine for their tumors. So they receive standard chemotherapy, which, as probably you know, it targets all the cells in our body that are undergoing proliferation. Either they are tumoral or not, the healthy ones as well. And these treatments are very, very aggressive. Well, we started by doing cell culture experiments in hormone sensitive, and the ones that are treated with tamoxifen, hormone sensitive breast cancer cells. And we compared the anti-proliferative effect of pure THC, which is represented in green, versus the THC-rich extract. And we represent here cell viability, the percentage of cells that remain alive in our cell cultures, versus different amounts of THC. And as you can see here, the more THC we add to the cells, the more cells we kill. That's the first observation, because the two curves go down. And the second observation is that the purple line goes below the green line. That means that we need less amount of the extract to produce the same anti-tumoral effect. In other words, that the extract is more potent than the pure compound, the pure THC. And this is for the hormone-sensitive breast cancer cells. What about HER2-positive breast cancer cells? Exactly the same thing. We also confirmed, and we knew this already, but we confirmed that HER2-positive breast cancer cells are also sensitive to cannabinoids. We can kill them with cannabinoids. And again, the purple line goes below the green line, meaning that it's more effective to use a whole plant extract than pure THC. And what about triple negative breast cancer cells? Exactly the same effect. And this is very important news because, as I said before, these patients don't have any kind of target therapy available right now. And what we see here is that we can potentially treat these patients with cannabinoids, because these cancer cells are also sensitive to these treatments. And again, the purple line is below the green line, suggesting one more time that it is a better option to use whole plant extracts than pure compounds. These experiments were performed in vitro, in cell cultures, but we have performed them also in vivo, in animal models of the 3 different subtypes of breast cancers. And I'm going to show you just one example, but the results have been the same in the 3 types we have analyzed. We have generated, for example, HER2-positive breast tumors by injecting this cell line in the back. Remember these experiments? In the back of these little mice. And we have treated them with either pure THC or a THC-rich extract. And we had another group treated with lapatinib, which is one of the medicines that these patients receive in the real clinical setting. And I want to say something about this slide. I never put doses in my slides because the doses we use for animals have nothing to do with humans. You cannot convert that. You cannot go to the internet and see how much a mouse weighs and then convert it to your weight. You can't do that because mice and humans are completely different. So it's not as easy. I wish it was, but it's not as easy as converting that. And that's why I usually don't put numbers in the slides, because I don't want anybody to go home, do the maths, and end up taking 3 grams of THC a day, which makes no sense, and it's going to make your life miserable for a few days. So that's why I don't put numbers. But in this case, I made the exception just for you to see that in these 2 groups, THC and the THC-rich extract, we are providing the animals the exact same amount of THC. Same amount of THC. The only difference between these 2 groups is that the extract comes with the other components of the plant. So it has THC, same amount as the THC group, but it also has terpenes and other cannabinoids. Very few cannabinoids, but terpenes are present there. So if we see a difference in the effect, it's not due to a difference in the amount of THC we are giving to the animals. It's due to the accompanying compounds. So that's the only reason why I put the numbers, and please forget the numbers right now. So what we did was measure the size of the tumors, and we observed, I'm going to go line by line. The white one corresponds to the growth of the tumors, the animals that did not receive any kind of treatment. The green line corresponds to the tumors that receive pure THC, single molecule. And as you can see, we observed a very small but significantly different effect in tumor growth. So THC decreased tumor growth to a certain extent. And the purple line is the effect that the THC-rich extract produce on tumor growth. And it's very easy to see that it is way more potent, as we saw in vitro, than the pure compound. And another thing that is very interesting is that the potency of the extract was the same as the one for lapatinib, a medicine that the patients are already taking. So we have something in our hands that is potentially as potent and less toxic than what patients are already getting. And I didn't bring the slides with me, but we had exactly the same results for hormone-sensitive tumors. They do respond in vivo as well to cannabinoid treatment, and the extract is more potent than the pure compound, and for triple-negative tumors as well. We did the experiments comparing the efficacy of THC versus the efficacy of the extract, and the extract was also more potent, and it was as potent as some of the standard chemo treatments that these patients are receiving. And by the way, in hormone-sensitive patients, in hormone-sensitive tumors in vivo, the extract was as potent as tamoxifen. I mean, if you have something with breast cancer around, you know what tamoxifen is. So we have something that is as potent as tamoxifen and less toxic than tamoxifen. So another question, can I combine cannabis with my other treatments, or do I have to quit my chemotherapy or my targeted therapies, my immune therapies? Well, we don't know, because no clinical studies have been conducted yet, so we don't have a definite answer for that question. But the preclinical information we have suggests that it's a very good idea not quitting standard treatments and combining them with cannabinoids. So the kind of therapy these cancer patients receive is very different, but we can group the antitumoral strategies in a handful of them. One would be what we call antimetabolites. So these are compounds that are very similar to the pieces that constitute our genetic material. They are called nucleotides, so we can provide the cancer cells with something that is very similar to the nucleotides, but they are slightly different. So when the genetic material is replicated, there are problems that make the cells commit suicide. So that's one of the strategies we can follow. And in this group, there are compounds like 5-FU. Again, these are very, very common medicines that a lot of cancer patients take. Capecitabine, gemcitabine, methotrexate, a lot of them. And there are examples in the scientific literature showing that combining this type of chemotherapy with cannabinoids provide better therapeutic results than using antimetabolites alone or cannabinoids alone. Here, they used a cell culture model of colorectal cancer, and they combined this compound, 5-FU, with another cannabinoid compound that activates CB1 receptor. THC would work the same way. And what they see, and here they represent, again, viability. So we want the bar to go down. And this is the effect that the antitumor compound produces alone, 5-FU. This is the effect that we get when we combine low doses of the antitumoral compound with cannabinoids. And when we go to a little bit more of this cannabinoid plus 5-FU, you see that the bars go down, down, down. They basically go to zero. That means that combining these two approaches, we are killing more cancer cells than when using each of them alone. Another group of anti-tumoral agents that are widely used in the clinic is what we call alkylating agents. So these molecules bind as well to the genetic material, to the DNA, and they cause problems in the replication process. And as a consequence, again, the cells end up dying. And we have things like carboplatin, carmustine, cisplatin in this group, and another alkylating agent that is called temozolomide. This is the one that was tested in the clinical trial promoted by GW, if you remember. So it's this compound. And we used it in the lab, and we observed striking results. This is tumor growth. This is an in vivo experiment, and the blue line represents how the tumors grow when the animals receive no treatment. This is the anti-tumor effect that THC produces. This is the anti-tumor effect that temozolomide produces. And this is what happens when you combine temozolomide and THC. The tumors don't grow, and some of them disappear. So again, the message is, we don't know if it's a good idea to quit standard therapies, we think it's not, or to combine it with chemotherapies, but the preclinical information we have so far suggests that combination is a better option. Same example or same results with what we call anti-mitotic agents. These agents don't target the genetic material, but the cytoskeleton, which is the scaffold we have within our cells, what gives shape, for example, to our cells. And sorry. Before a cancer cell duplicates, she has to rearrange this structure, the cytoskeleton. So we can target that process as well. Not only target the DNA material, but also we can target the cytoskeleton. And we have a lot of compounds that do that: paclitaxel, docetaxel, taxols in general, and a lot of other compounds. And there are a couple of examples in the literature showing that combining this type of compounds with cannabinoids is, again, a better option than using either of them alone. Here, they represented the results the other way around. We want the bar to go up because we are representing live cells, or alive cells, not dead cells. So we want the bars to go up. And this is the effect that endocannabinoids or the paclitaxel alone produce, and when we combine them, we get the best results. So again, it seems that combining chemo and cannabinoids is a very good option. What about radiotherapy? Because a lot of cancer patients and a lot of other cancer patients, breast cancer patients and non-breast cancer patients, they receive radiotherapy also. Well, there are experiments, preclinical experiments, showing as well that combining these two approaches is a better idea. This is tumor volume in these graphs. This is a model of glioblastoma, the brain tumor I've been talking about. And this is how the tumors grow when the animals don't receive any treatment. This is the behavior when they receive cannabinoids, and this was a mixture of THC and CBD. This is the effect with irradiation. And this is how the tumors grow when the animals receive both radiation and cannabinoids. And the line is here. So the tumors basically don't grow. So all these results are suggesting that people shouldn't quit standard therapies, and that maybe combining them with cannabinoids is a better idea. And what about patients, actual patients, not mice? Do we know anything about combination of therapies? Well, the only study that was designed specifically to answer that question was the one I mentioned before, that was performed in the UK and Germany. We don't have the results yet. I cannot make any comments on that. But we do have information coming from other studies that suggest that combination of chemotherapy or radiotherapy with cannabinoids is not a problem. And hundreds of studies have been conducted in cancer patients for pain, for example. The goal was not analyzing if the anti-tumoral effect was better or was improved. The goal was testing if the cannabis products worked for pain. But these patients were undergoing their regular anti-cancer treatments. So if any kind of negative interaction between cannabinoids and their therapies had occurred, we would know. And in any of these studies, nobody mentioned any negative interaction between treatments. So we can suggest that we shouldn't expect any kind of negative interaction between these treatments. There are some things that we have to keep in mind that we can discuss later if you want, but in general, and although no clinical studies have been conducted to answer specifically this question, I think we have pretty much, we have a lot of information suggesting that that's the case. So there are many other questions that patients ask us. What cannabis strain, that's a typo there. This is a strain. What cannabis strain do I use? Which THC CBD ratio is the best for my type of cancer? How much do I use? How do I take it? Suppositories or administration? And I don't have time to go through these questions now. Mara Gordon, who's speaking next, she will give you more information about this. But the warning sign is to remind you that there are a lot of people out there that are looking forward to helping patients, but there are a lot of people out there that are looking forward to making money out of patients. So you have to be very careful and go, I mean, my advice would be go to the original sources of information. Facebook is fantastic for support groups and things like that, and as a start point. But if you are looking for questions, answers to these questions, my advice would be going to the original sources of information, because, I mean, there are dangerous people out there and dangerous messages out there for the patients. And just to summarize, the conclusions would be that we have a tool in our hands that works as an anti-tumoral tool, at least in preclinical models of cancer. We know also that the whole plant extracts work better than the pure compounds. These tools are safe, and we know that not only from the preclinical, but also from the clinical setting. And we also know that they synergize very well with the standard anti-cancer treatments. And we know, and let's not forget this, that these compounds have additional advantages that are very good for patients, for cancer patients. They are analgesic compounds, they stimulate appetite, antiemetic, and many more. So what do we do next? Well, we are a group, we are basic scientists, we are biochemists, and our work is asking questions and getting answers. And we have worked for many more years. We have many questions that we want to answer. We want to know more about the mechanism of action of these compounds in cancer cells. We want to know more about how they combine with other compounds. The more information we know, the better we can attack the cancer cells. And I mean, we have worked for years. But we also need, and we need it immediately, clinical information, because we have to make this thing accessible to the patients. And we can wait, and we will have to wait for the clinical trials like the one I showed you on the slide, but that will give us an answer in 20 years. And in the meantime, what do we do? So we have to get information from wherever it is available. And in places like this, or in my country where medicinal cannabis programs will be implemented soon, hopefully, we have to get all that clinical information. If there are people here that are going to start selling products to patients, why don't you collect the information about the patients? Why don't you do a patient follow-up and share that information with people that can convert that information into graphs to show the doctors and convince them that this is an additional tool that they can use for the patients? Well, thank you very much. Questions?
I think that's yes.
Is it time for questions?
Thank you very much for this.
Oh, thank you. Thank you very much. Thank you.
Działa. Proszę Państwa, rzeczywiście troszkę mało czasu jest, no ale nie wyobrażam sobie, żebyśmy przynajmniej kilku pytań nie przyjęli i nie odpowiedzieli. Mogą to być pytania, mogą to być questions, mogą to być preguntas.
I will try to speak English. I'll try to speak English here. I want to examined in your work. Was it CO2 or ethanol extraction?
Yeah, thank you for the question, because I think it's very important. The extract was produced by Onceldes, the owner of the company is sitting here and she's going to talk right now. But the answer is ethanol extraction. And this is one of the things I'm going to take the advantage of, following your question answer. The warning sign that I put in one of my last slides has to do with that as well. There are tons of tutorials out there showing you how to produce extracts, and they suggest you to use things like naphtha, like hexane, like isopropyl alcohol. Those things are very toxic, very, very toxic, not only for manipulation, not only for the person that makes the extractions, but also for the patients. In all of these tutorials, they say that you have to evaporate the solvent, but there are some residues over there that will not disappear, and you are giving poison to the patients if you are giving these products. So the answer to the question was ethanol extraction, which we think is the best and only option right now for this, the best option for these products. And please be careful with the tutorials out there. Well, I'm not an expert in...
Przepraszam, un segundo. Pytanie było, dlaczego nie ekstrakcję nad krytycznym dwutlenkiem węgla? Porque no CO2.
Okay. Yeah, I'm not an expert in extraction processes. As far as I know, well, Mara will give you a better answer to the question, but what I can say is that CO2 extraction, first of all, requires very expensive equipment, which is not available, accessible for many people. And also the yield of terpene extraction is not as good as with ethanol. But I would prefer another person to answer this question. So I would suggest you ask Mara when I finish.
I have two questions. Firstly, sorry for my English because it's not perfect. Okay, so you spoke about breast cancer, brain tumors, and what about blood cancers? I mean, leukemia. What do you have some experience?
Yeah, that's a very interesting question. Yeah, they seem to respond. They seem to respond like solid tumors. Yeah, we still don't know what they have in common, cancer cells, that make them responsive to cannabinoids. But hematological tumors, they respond exactly the same. And in this case, there is one study performed in patients, in actual patients. It was not patients exactly, but it was samples taken from patients. And cannabinoids kill these type of cancer cells, and they prevent the progression of the disease. Yeah.
Okay, and also the patient with brain tumor, for example, astrocytoma. The patient, for example, had an operation to cut out the tumor. And what is your opinion? Is it correct to give them THC to protect? The tumor doesn't grow up. What is your opinion?
Well, that's an extremely difficult to answer question. I'm going to try to start answering from the bench, from what we know from the lab. And we have always performed our in vivo studies once the tumor is established. A lot of people ask us, is there any way we can prevent tumor appearance by using cannabis before we have a tumoral disease? And we don't know. We've never done these experiments in the lab because we wanted to make the experiments as similar to the human pathology as possible. But after 20 years, I think we know enough to maybe try if we can prevent this disease by using cannabis in advance. We don't know. And regarding actual patients, well, that's difficult to answer because, again, we don't have controlled studies. But there are a few things that we have to keep in mind when trying to decide whether to combine standard treatments with cannabinoids. And one of them would be checking the potential interaction of these drugs with a protein that is called cytochrome P450. This is a protein that is in charge of metabolizing a lot of toxic compounds that we get from the outside, including medicines. And CBD specifically can inhibit this protein. So that's something we have to keep in mind. It's not that you can't use two compounds when one of them is affecting the cytochrome P450. It's just that we have to be more careful with the doses. So you have to talk to your doctor, ideally, and tell him that you're using cannabis and that you may have some changes in the actual amount of the other medicine that you have in your body. And there are a few things that you have to keep in mind, but in general, if I was in your place, I would use it. That's the only thing I could say, because the risks are very low. So it's not because the efficacy is going to be high, it's because we don't know, but the risks are low. So, and that's something we always have to keep very present, that we are not talking about a medicine that if we don't use correctly, we're going to kill someone. So that's one advantage that we have and that we have to keep in mind. Yeah.
I would like to ask you about antioxidants. Is it correct to take high doses of vitamins with THC? Hello.
I'm sorry. Antioxidant, that's a very good question too, and that's a tricky one because we know, not for THC, but for CBD, the main mechanism of action, of antitumoral action, is the generation of oxidative stress. And antioxidants prevent oxidative stress. So if we are combining antitumoral treatments that produce oxidative stress, and a lot of them has that as a mechanism of action, with CBD that prevents the generation of oxidative stress, we may be having opposite things. We may be doing opposite things. So we don't know. We really don't know. But in this case, if the antitumoral therapy is specifically an antioxidant therapy, I would recommend to be cautious with the use of CBD in particular. But still, we don't know. And it's interesting because, for example, the effect of CBD in cancer and non-cancer cells is completely opposite regarding the generation of reactive oxygen species. In cancer cells, CBD produces oxidative stress. And in non-cancer cells, CBD protects from oxidative stress. So we don't really know what would happen in actual patients when combining CBD with anti-cancer treatments that trigger oxidative stress. We don't know. Sorry, I can be more precise.
Could you please respond to the Rick Simpson treatment? How much is known about it? Is there any evidence for assistance?
Yeah. I'm so happy that you're asking me this question because the warning sign is mainly due to this whole Rick Simpson protocol in theory. We receive so many mails from patients saying that they are following the protocol that says that you have to use 1 gram a day of cannabis for 3 months and your tumor is gone. And that has absolutely no scientific rationale behind it. None. If you think about it, think about what cancer is. Cancer is 200 different pathologies. Does anybody really think that we are going to be able to treat a pathology that is 200 different things with one thing? Just to start. And what is the meaning of 1 gram of cannabis? What is in 1 gram of cannabis? What type of cannabis? Saying 1 gram of cannabis is saying nothing. It's no detail. How much THC is in there? How much CBD is in there? Why 3 months? It comes from it has no scientific background whatsoever. So, no. Rick Simpson, I'm sorry to say this out loud, but I'm not a big fan of anything related to Rick Simpson because there is no scientific rationale behind it.
Ja też mam pytanie.
Hold on a second.
Ja też mam pytanie trochę może na inny temat, czyli chodzi o choroby z autoagresji. Jak tutaj kanabinoidy mogłyby zadziałać, czy w ogóle jakieś były badania robiły w tym kierunku? Z chorobami autoimmunologicznymi. Chorobach.
Okay, that's not my field of expertise, but there are some autoimmune diseases for which cannabinoids have been proved to be very effective. Again, I will suggest you to ask Mara Gordon because she has experience with actual patients and she can share what she knows with you.
Sí, ¿me podría decir si hay alguna diferencia, alguna valoración sobre el uso de los diferentes extractos de cannabis o bien simplemente la inhalación?
Przepraszam, może się wtrącę, bo pewnie nie przewidzieliśmy tej możliwości. Pytanie było o różne rodzaje ekstraktów versus po prostu wdychanie zioła, tak. In English, por favor.
Okay. So, difference between routes of administration, basically, right? So that's another one of my favorite questions too. It depends on what your goal is. The different routes of administration are associated to different times where the effect peaks. So the effects can be faster or slower depending on the route of administration. And also with respect to the time the cannabinoids remain in your body. So in inhalation, for example, you have a very high peak, very, very fast in your blood, but it disappears very, very fast as well. Is this interesting therapeutically speaking? It may be for some things. For example, if you're a patient that has pain and a pain that you have to get rid of immediately, this is the perfect way to use cannabis, for example. If you're a cancer patient and what you're looking for is an anti-tumoral effect, you don't need or you don't want to be inhaling every 2 hours. That's very uncomfortable. So you need another route of administration that allows you to have high levels of cannabinoids in your blood for prolonged periods of time. And that's better achieved by oral administration or sublingual administration. And I'm going to talk about suppositories because this is another route of administration that a lot of patients are considering now because they say that they can use huge amounts of THC per day without having any psychotropic effects. And that's absolutely true. They can use suppositories with up to 1 gram of THC per dose, and they can use a couple of them a day, and they don't feel anything. And they don't feel anything because the THC is not getting to the blood. Because the absorption of lipids through the rectal mucosa, and cannabinoids are lipids, it's very, very low. So they are wasting the products, in other words. The cannabinoids are not getting to the bloodstream, and that's why they don't feel anything in the brain, because the CB1 receptors that I showed you before in the slide where the slice of rat brain was stained in black, these receptors are not getting activated because the THC is not getting there. And if the THC is not getting there, it's not getting to the tumor cells either. So that's why suppositories, as far as we know now, are not a very good option for patients. Unless you have something very local, or you have the tumor right there, or it's something that requires very low penetration.
Rozumiem, że Dorota chce dopytać a propos czopków.
Tak, pani profesor, ja chcę faktycznie dopytać co do czopków, dlatego że od kilku lat to...
Not working. Can you start again first?
Tak, Jeszcze raz zacznę. Chcę dopytać na temat czopków, ponieważ od kilku lat stosujemy czopki u naszego dziecka z CBD, nie z THC, co prawda. Więc pierwsze moje pytanie, czy przez śluzówkę odbytu nie wchłania się tylko THC, czy wszystko, bo to by przeczyło jakby doświadczeniom. A po drugie, jaka jest wobec tego różnica w śluzówce jamy ustnej, a śluzówce odbytu, bo rozumiem, że chodzi o wchłanianie się naczyniami włosowatymi. I ostatnie pytanie jakby z tej grupy, to czy ten efekt psychoaktywny, który nie występuje poprzez podanie per rectum, nie ma związku z metabolizmem wątrobowym, bo przecież jak się podaje przez śluzówkę, to następuje pominięcie pierwszego przejścia przez wątrobę. Tak nam to tłumaczono i w wielu wykładach jakby była poruszana kwestia, że przez śluzówkę jest to doskonała metoda, więc jak to jest?
So my answer is, regarding CBD and THC are very similar, chemically speaking. So the same problem that THC has, CBD has. And if it's working for your kid, keep using it. I just wonder how much CBD would you need if you would administer the compound orally. It would be probably less than what you're using now. But if it's working, I mean, keep going. That would be my question, my answer regarding CBD and THC. But the problem is the same. Chemically speaking, they are very, very similar. So THC doesn't get to the bloodstream, CBD doesn't get either. I mean, it gets, but in very, very low amounts. So I think you could save a lot of money and product by using orally. But that's my guess. So, yeah, under the tongue. Yeah, that would be...
No tak, ale właśnie o to pytałam, czy śluzówka jamy ustnej czymś się różni od śluzówki odbytu, bo śluzówka to śluzówka, z tego co mnie uczono przynajmniej.
It's different. It's completely different. It's completely different. The absorption is completely different. Yeah. The huge blood vessels that you have under the tongue, they have a very thin layer, and the lips go there very well. And the rectal mucosa, the structure is completely different. Yeah. And then the other thing you mentioned about going or not through the hepatic system is true, but that's not the cause of the lack of psychoactivity. What is produced when THC is processed by the liver, some of those compounds are even more psychoactive than THC. So that's not the explanation for the lack of psychoactivity. It's just because you are not getting the product to the brain.
Okay, good afternoon. I have a question, the conclusion from your lecture, because in oncology the best route of administration is orally, or in special cancers it could be the inhalation or another route of administration, where the mode of action is the best, in what kind of route of administration?
It depends on your goal. If you want to have immediate relief, inhalation is the best option, but it lasts very few hours. And if you need high amounts of cannabinoids for a long time, oral administration would be better. So it depends on what your goal is, because the bioavailability is different, the pharmacokinetics is different, so it just depends on what you want.
I want to, for example, reduce the growth of the tumor. So the best is oral administration, yes?
Yeah, because that way you would have, I mean, it will take longer to have high levels of the cannabinoid, but then they will last high longer than if you use it by inhalation. Yeah. I don't have it here with me, but I have a graph where I compare. If you use inhalation, the peak will be like this. If you use oral administration, it will take longer to peak, but it will last longer. So it's a completely different kinetics.
And what dosage, about what dosage we talk about in the oncology, if you can say?
No, I can't say. If you want, you can try to ask Mara, but she won't answer either. That depends on so many things, so you have to talk to her in private.
I have one question. One question regarding the concentrate of this THC that can be helpful.
Say that again, sorry.
The concentration, the level of the THC.
The concentration?
Percentage. 50% of THC.
Okay, that's not important. What is important is how much THC you're getting to the patient and how much in milligrams, in mass. Those milligrams can come from something that is very, very concentrated, and then you have to give him a very small amount, or it can come from something that is very diluted, and then you have to give the patient a huge amount. But the percentage is irrelevant. It's like, for example, taking coffee in the morning. Imagine that you need 50 milligrams of caffeine to be fully functional in the morning. You can have those 50 milligrams of caffeine by taking one espresso or by taking, I'm not a coffee drinker, but 5 coffees with milk. The amount of caffeine you take at the end of the day is the same, but one comes from a very concentrated source and the other comes from a very diluted source. So the percentage is irrelevant. What you have to know and you have to ask to your provider is how much THC and CBD you have in your medicine. That's the key question. In milligrams per milliliters. Okay, no percentage.
Did you perform study to compare CBD or to THC in the therapy anti-tumor, especially in the animals model or in vivo?
You're asking if we compare the effect of THC with that of CBD. Okay, yeah. There are many studies conducted with CBD, and it is also very effective in producing anti-tumoral responses. Less work has been done, and we know a little less about the mechanism of action, but it is also a very effective anti-tumoral compound. But it works better in combination with THC.
Jacek, poczekaj. Jacek.
In animals model or in vivo models?
Yeah, in both, in cell cultures and in vivo. We have done experiments with CBD in models of breast cancer, and it works very, very well. And other groups have done the same experiments in glioblastoma models, in skin cancer models, in pancreatic cancer models. So a lot of studies have been conducted with CBD also in vivo, and it works. But again, it works better in combination with THC.
Proszę Państwa, ja myślę, że pewnie jeszcze parę niezaspokojonych, co najmniej parę niezaspokojonych rąk byłoby w górze, ale myślę, że przejdziemy do następnego prelegenta. Tym bardziej, że pytania, które się tutaj pojawiły, kierowane do pani profesor Sanchez, właściwie niektóre z nich powinny być do następnej prelegentki i skierowane, także proszę je trzymać w zanadrzu. Myślę, że zostaną, przynajmniej duża część z nich będzie mogła znaleźć odpowiedź.
Bardzo dziękujemy za wykład, pani profesor.